We look at immunological techniques and applications part 2. We will start with enzyme-linked immunosorbent assay, what we call ELISA, which is a very sensitive immunochemical technique that we normally use to determine the presence of a specific antigen or a specific antibody in a given sample, maybe a blood sample, and also we are able to quantify how much of that antigen or antibody. We can also call it solid phase enzyme immunoassay, and it employs an enzyme linked antigen or enzyme linked antibody as a marker for detection so that there will be a color change as we shall be seeing. So an enzyme is usually conjugated to the antibody, which now reacts with a colorless substrate, um, and this generates a colored reaction product. So we do have a number of enzymes that have been used in the ELISA technique, including alkaline phosphatase, horseradish peroxidase, beta-galactosidase, and an example of the substrate for horseradish peroxidase is the 3355 tetramethylbenzidine. The principle of ELISA usually will happen in 3 combined steps. One of them is the immune reaction, which is why we are calling it an immunological technique. So we have the antigen antibody reaction. Then we have the enzymatic chemical reaction, because we said that we normally have an enzyme tagged to your antibody. So that enzyme catalyzes now a substrate which is usually colorless to give us a chromogenic product or a colored product. Then we have quantification and signal detection, which is now the measurement of the color intensity, which is synonymous to how much of the antibody or antigen that was your analyte of choice. So the general procedure for ELISA will start with coating. Usually we will have a microtiter plate like this one here. We have such a plate. So within these wells, we normally coat. Now you can coat with your antigen or antibody, and then we normally do a wash step, and then we do what we call blocking, so that areas where we don't have the antigen or antibody then can have some protein that is neutral that does not react, so that now it is able to block to provide unspecific binding. And then once you have washed off the excess blocking reagent, then we normally add your antibody if you coated with antigen. If you coated with antibody, then you're going to add your antigen. And then you usually, if you have coated with antigen, you add an antibody that is conjugated to your enzyme. And now that is what, after you have added your substrate, you're able to get now a chromogenic reaction product, like for example blue, which now the intensity of this color is going to give us the concentration of your analyte. So we look at the different types of ELISA and how the procedure looks like in a snapshot. So we do have what we call direct ELISA, which is where we have only one set of antigens and we have only one set of antibodies. And in this case, usually is where you will now coat your well. So for example, here you have coated your well with the antigen of choice, and now you add an antibody which is conjugated to the enzyme, and then you'll add the substrate for chromogenic color reaction. Yeah, so this is a direct ELISA. So in this case, this antigen, because it's just one, will actually be now the antigen that is within your serum sample. Yeah, so that your patient's blood sample, you will now coat your plate maybe overnight to allow the antigens to bind, and then the next day you're able to add your antibodies. So this step directly is very quick, and also what we call cross reactivity, because we have other types of ELISA that utilize another secondary antibody in addition to this primary antibody. So in this step, because it's direct, only one antibody, you prevent any cross reactivity that may happen with other steps. But this step, as much as we're able to prevent cross-reactivity, the immunoreactivity of this primary antibody is a bit hindered because it is labeled. Yeah, so you will realize that when you have a primary antibody that is not labeled, it will maximally be able to bind onto the antigen. So in this case, immunoreactivity is low. That tells you that the signal amplification is also low, and therefore may not be able to also detect very minute or very little amounts of your antigen if you're testing a blood or a serum sample. Then labeling of every primary antibody every time you want to do an assay in the lab will become time consuming and expensive. We will have other tests that you can easily use the same secondary antibody for different assays. Yeah, and then it is not very flexible because the choice of primary antibody label is different from one test to another. Then we have indirect. Yeah, so in this cartoon diagram, the red is the marker protein. Then we have the primary antibody. Then we have a secondary antibody that is conjugated to the enzyme, which looks yellow. So the first step here is, of course, you add your sample, then you incubate to ensure that your antigens or proteins are bound to the well. Then you block. Usually we have a wash step to remove what has not bound. Then you block with a neutral protein like milk proteins like casein, BSA. Once you have blocked all the regions that don't have any protein, then you normally also wash. And after that, now you add your primary antibody here. So your primary antibody, we expect it to bind to your antigens or proteins. Then you do a wash step to remove any unbound antibodies. Then you add your secondary antibody, which now binds to the primary antibody. Yeah. And then you do a wash to also remove unbound secondary antibody. And then now you can add your substrate so that now the enzyme on your secondary antibody will catalyze the substrate. And now in this case, the reaction turns blue. So these are all the steps that I have said now in text form, and you can appreciate that by reading through up to 9. So what happens is that now you have gotten your color reaction or your colored endpoint reaction. So what you do now, you can be able to read this using an ELISA reader, and using that you can also be able to quantify. Normally once you do an ELISA reader, you get optical densities. So now the optical densities can now be converted to concentration using standard curves. Advantages of this is that you can have a wide variety of labeled secondary antibodies available commercially so that you don't have to design your own. Because usually if your primary antibody is maybe a goat antibody, you can get any other anti-goat antibodies that are now conjugated to the enzyme, and they can be easily available because you don't have to design each time. Then the same labeled secondary antibody can be used for a variety. That's what I meant, that you can also use it for a variety of primary antibodies. Then maximum immunoreactivity of the primary antibody is retained because it's not labeled to an enzyme. Then sensitivity definitely will be increased because the primary antibody has several epitopes in which the secondary antibody can be able to bind. Therefore, the signal can also be amplified. Cross-reactivity definitely might occur. That's a disadvantage. So the fact that we can use several secondary antibodies means that we could end up having some cross-reactivity, which may result to non-specific signal. Then an extra incubation step, you have seen that this one has several. In addition to just the first incubation, we have now another because we have two antibodies. So that makes the experiment a bit longer. Then we have what we call sandwich ELISA. Now this one, again, still all of these tests usually determine the specific antigens. And in this case, what we do is that our wells are coated with a capture antibody. Yeah, so in this one, this is what is different about all of the other with the rest. This one you have to coat your microtiter plate with capture antibodies. And then now from your patient sample, you add your antigen. So we're expecting that your patient sample has antigen that is specific to this capture antibody. and then you add another antibody, which is now your primary antibody. And once you use your primary antibody, it is also going to recognize another epitope on your antigen. And of course, finally, you add your secondary antibody. So the beauty with this test, as in this other diagram here, is that you can do this both directly or indirectly. So we do have what we call direct ELISA and indirect ELISA. So after you have coated with a capture antibody on your plate, you have added your antigen. So if you do direct ELISA, the antibody that you add here is going to be labeled to the enzyme, so that now you just add your substrate and there'll be the signal. If you do it indirectly, it's like the one I have just showed you before. You have your capture antibody, you have added your sample which has the antigen, then you have a primary antibody that is not conjugated to anything but is able to identify your antigen. Then you add a secondary antibody that is specific for the primary antibody, and then of course you'll add the chromogenic substrate to have now the reaction product. So this particular test is very highly specific because the antigen is specifically captured and detected. So you can see there are two antibodies identifying the same antigen. That makes it very specific. And if it is very specific, it is then good for especially the diagnosing disease, because you won't miss out if somebody has an infection. It is suitable for complex or crude or impure samples which have many antigens. So by having two, you prevent any cross reactivity, so that if this capture antibodies may have detected an antigen which does not have an epitope for the primary antibody, that means that at least you're able to remove any impurities within that sample. It is flexible and sensitive, and we have said that it is both usable as a direct and indirect, which I have demonstrated using this diagram. Now we go to blotting techniques. There are many, but we will look at just one. So blotting just means a method of transferring DNA. You can transfer DNA, RNA, and proteins onto a carrier membrane so that it can be separated. And this also usually will follow what we call gel electrophoresis. We have three different types of blotting techniques. We have southern blotting, northern blotting, and western blotting. Southern is used to detect DNA, northern for RNA, and western blotting for protein. So we look at western blotting, which is now the technique we have said for identifying proteins that have been separated by electrophoresis. And in this case, we have to use antibodies to target those particular proteins. So what happens is that because blotting means to transfer, you run your gel electrophoresis, which separates your proteins within that particular sample dependent on size. Yeah, so that remember we also have a ladder within the gel electrophoresis which gives us the molecular weight. So gel electrophoresis will separate our proteins dependent on their size and also on their charge. After that, we need to transfer what has separated onto another membrane, and that is why this setup is important. So you have your transfer buffer in a trough, then you have some wrap, then we have a Watman paper that you normally have in the lab, the white ones. Then now here the purple one is critical. This is your gel, the one that you have already separated. So you want the proteins within that gel to transfer to a membrane. That is the membrane that you put on top of the gel. This membrane can be like nitrocellulose, it can be PVDF. So you put the membrane there, and then another Watman paper, and then now you have your paper towel stack. You stack paper towel, yeah, and then you add a weight on top because the weight is going to help press down. So once you have done that, you have transferred um you can also do this is manual. We also have an electronic way where you use electric current to transfer from gel to membrane. So depending on what is there within your lab, either manual or electronic. Then after you have transferred, now you have your membrane. You block, just like we are blocking in ELISA. We block any areas that don't have any protein to avoid unspecific binding. So blocking, we can use milk proteins like casein and all that. Then after you have blocked, then we normally add our primary antibody. So this primary antibody is not tagged to anything. You expect it to bind to your protein of interest. Yeah. And then now you incubate, and then after that, add your secondary antibody, antibody 2, which recognizes your very first primary antibody. Now, this secondary antibody can be radiolabeled. Remember, we talked about radiolabeling. We can also have it bound to an enzyme, which we call a reporter enzyme. We can also use fluorescent. We can have it tagged or labeled to a fluorophore, so that now you can either use auto radiography, you can use fluorescent microscopy, you can also visualize using naked eye, that is chromogenic or change in color. Yeah, so this is just a setup. You have your target protein, you have, so this is your membrane that has your transferred proteins. So your antibody will target, for example, this protein that is in this region. So that tells you that we can be able to tell the weight depending on how far it has moved on the membrane. Then we add your primary antibody, then another antibody which is conjugated to an enzyme for us to have the signal. Yeah, this is the same way. Again, you have done your SDS page, gel electrophoresis, done the blotting on nitrocellulose, labeled. You can also do direct. You can see now here we have direct primary antibody conjugated to your enzyme. And you can now be able to see that particular protein. So you can be able to identify specific proteins, identify their mass or molecular weight, and this has been used a lot as a confirmatory test for HIV by using anti-HIV antibodies. We can also use it for testing BSC, commonly referred to as the mad cow disease, and also some forms of Lyme disease. Now, another test we can do using immunology is immunoelectron microscopy. We talked about electron microscopy. So here we are adding now within electron microscopy, the transmission or the scanning electron microscopy, we add the use of an antibody to locate certain proteins within the cell. So we have several cellular events that can be just missed if you do just ultrastructural studies where you observe the position of the cell, organelles, and all proteins, etc. So if we now add immune or localizing of proteins using antibodies within cells and tissues, it becomes very practical for also helping us to diagnose disease. So this technique uses antibodies to detect intracellular location of structures, particularly maybe proteins, so that you make ultrasections that then you label them with antibodies against the required antigen. And then these antibodies, in this case, because you're using electron microscopy, we label them with gold nanoparticles. So this is just a review of what we have already said about scanning, which gives us the surface topography, and we have the transmission, which gives us the ultrastructural images of our tissues and cells. So this is the pictorial. So you have your ultrastructural image, you raise an antibody against, let's say, this antigen, and your primary antibody, then you added a gold conjugated secondary antibody. So the reason for gold, or what we call immunogold labeling, is that colloidal gold are most often attached to secondary antibodies, which in turn are attached to the primary antibodies that has bound to your specific antigen. Gold is used because it has high electron density and therefore increases electron scatter, so that it can give us high contrast, because usually these images, most of them will be black and white. So the contrast of very dark spots we can be able to achieve with gold nanoparticles. But you can also do immunostaining, what we call immunohistochemistry, where you just use color or fluorescence, and you can be able to also visualize under maybe a fluorescent microscope. So the reason why this is important, of course, it's powerful. Using immunogold is a powerful tool to help us locate different proteins and antigens, how they're distributed within an ultrastructural level. And then it is a powerful technique for identifying active sites and also presence of biomarkers. And then primary antibodies designed to bind to specific antigens. And when you do gold conjugation, then that probe is excellent for electron scattering. That becomes very nice and better even than immunohistochemistry. This is actually what I have somehow said, that immunofluorescence now is where the antibody, whether directly, where you just have one antibody targeting a certain antigen, is bound to a fluorophore, or you can have the indirect, where we have the antigen, we have the primary antibody, then we have these secondary antibodies against the first antibody bound to a fluorophore. And what happens is that once you have that, then you use an electron microscope. And this electron microscope is able to give you images like the ones you can see here, if your antigen of choice was present within that particular tissue. So this is also a very good technique where we use immunofluorescence. And this can also, is just that this now using tissues. When we talked about flow cytometry, now we are using immunofluorescence, but now in the situation of a cell, one cell at a time, when you are trying to identify uh, particular cells, maybe CD4 cells, other immune cells, or any particulate particle, we can be able to visualize using immunofluorescence. So you can have both for tissue and for cell. So that is it. If you have any questions, you can email